phosphoenolpyruvate, for example, and you can convert it into erythrose, which is then a precursor for the aromatic amino acids, the phenylalanine, the tyrosine, the tryptophan. You can take pyruvate and convert it into alanine and lysine and isoleucine. You could take acetyl-CoA and convert it into valine and leucine. Okay, so the idea is that these pathways are not just specific to catabolism. And when I say that they provide carbon skeletons, I'm referring to these intermediates that can be taken away from these pathways to synthesize things instead of break them down. Yes, so carbon skeleton is that 6-carbon glucose sugar, yes. Okay, so I hope that makes sense. If there are any questions, please let me know. Notice you can make aminos acetyl-CoA to synthesize fatty acids. You have all of these various amino acids that are possible. The restriction, the limitation is that microbe must have enzymes that allow, each of these steps to be carried out. Conversion of serine into glycine has to be carried out by enzymatic pathways. And so if microbe lacks those pathways, then it can't carry that step out. If it possesses those pathways, then it can carry out that step. Okay, so generally microbes are going to be producing a lot of these nutrients themselves, so they're going to have a lot of these diverse pathways, or they're going to live in a community where they can interact with one another in a sort of mutualistic way and benefit from each other's abilities. Okay. All right, so going back to that idea of that we can use these very central pathways for synthesis purposes as well, not just for catabolic purposes. And as I said, I'm just going to give you one example so that you understand. And like I've given you these examples that you don't have to continue to break them down all the way down to CO2. You can take them out of these pathways and channel them to synthesis pathways, right? So here we have the EMP pathway, right, the Embden-Meyerhof pathway, which we generally refer to as the glycolysis pathway, which can be reversed. So the steps can be reversed so that you can synthesize glucose. So if you look at these steps initially, notice some of these steps were represented with bidirectional arrows, and so those steps are freely reversible, right? So only the concentration is driving which direction they will be moving in. If you have a lot of 3-phosphoglycerate, it will move forward to make 2-phosphoglycerate, but if you have a lot of 2-phosphoglycerate and very little of 3-phosphoglycerate, then the rate of reaction would reverse, okay? Or the direction of the reaction, I should say, it should reverse. So there are some steps that are freely reversible, there are others that are not, and that is how the microbe would control, which direction to carry these reactions and the forward direction, so that you break glucose down into pyruvate. Or what if you don't have glucose and you're growing in presence of proteins, and there are certain amino acids that could be broken down to produce pyruvate, right? For example, alanine, lysine, isoleucine, valine, leucine, all the amino acids that will be very commonly present in any protein. So we can break down those amino acids, convert them into pyruvate or something else and convert it into other intermediates, right? But let's say we start off with pyruvate, and you have to express this enzyme pyruvate carboxylase to convert it back into phosphoenolpyruvate. And in fact, there's an additional step. Okay, first it has to be converted into oxaloacetate, which doesn't happen when we were looking at the glycolytic pathway leading up to synthesis of pyruvate. But when you try to convert it back into phosphoenolpyruvate, there's actually a couple of steps, both carried out by unique enzymes. Okay, and so these enzymes must be present and expressed when needed in order for a microbe to convert these pathways backwards. And so the remaining reactions would occur with the same enzymes as we've discussed. And then eventually you have the phosphofructokinase step. Okay, notice this is being now carried out by a different enzyme, fructose bisphosphate, TASE. Okay, so that it's going to remove the phosphates instead of adding the phosphates. And then you again have the glucose 6-phosphatase, which is again going to remove the phosphate instead of adding the phosphate. So on this side, you're seeing the reactions that occur in glycolysis okay, that require ATP, right? And on the other side, you're looking at reactions that refer to what we refer to as a process called gluconeogenesis, okay, de novo synthesis of glucose starting from non-glucose sources. And so you have, you know, different enzymes at 4 of these steps, right, that are going to have their own requirements. Pyruvate carboxylase requires ATP, but phosphoenolpyruvate carboxy kinase requires GTP, right? So they have their own requirements. Okay, you have another step that requires input of ATP. Then you have, the requirement of NADPH actually, which, it's, I guess they're showing it as NADH here, but, you're going to have to provide electrons that were removed. Okay, so during glycolysis, you took off electrons. During gluconeogenesis, you're going to, at those steps, add those electrons back, and that is why you need that electron source, or NADPH, okay? And so you have some reactions that are freely reversible, other reactions that are irreversible. They must be carried out by distinct enzymes, and by controlling the expression of those distinct enzymes, microbes can decide which direction to carry out these reactions in. Okay, so they're not kind of confused and just randomly doing whatever they want to. If they have plenty of glucose, none of these enzymes would be synthesized, okay, because they have plenty of it and they can use it to synthesize whatever else they need. However, if they don't have glucose, then they can start to express these and turn these 3 off, okay, so that they don't break down the glucose the cell is synthesizing via this reversal of the pathway via gluconeogenesis. Okay, so just having these various steps being controlled by distinct enzymes makes the same enzymatic pathways, distinct for either breakdown of glucose via glycolysis or synthesis of glucose via gluconeogenesis. Okay, questions? Okay, all right, so that's the EMP pathway. Now let's look at another pathway, which is found in bacteria. And so we have the ED pathway. And in the ED pathway, like I said, not all organisms would have it. Our cells don't have it. And so we start off with glucose, and it's converted into glucose 6-phosphate, so that step is the same. But instead of converting that into fructose phosphate and then fructose bisphosphate, you're going to convert it into 6-phosphogluconate. So this is a step that is unique to ED, okay? And then notice it's going to generate a different type of a electron carrier, NADP, okay, was converted into NADPH. So you have this, oxidation reduction step where glucose 6-phosphate is being oxidized and NADP is being reduced. And these are electrons that can be channeled into amphibolic reactions like gluconeogenesis, okay, or synthesis of amino acids, okay. And so you have that synthesis that you did not find in EMP pathways. So that makes this pathway unique, that if a cell has high need for electrons for synthesis of say sugars and things like that, then they could, well, not sugars, maybe amino acids and like lipids and things like that, they could rely on this pathway to produce those, source of electrons that are not going to be channeled into electron transport chain, that are not going to be used for ATP synthesis, but rather channeled into amphibolic pathways, so complex molecules could be synthesized, okay? And then you have the conversion of that phosphogluconate into a compound called 2-keto-3-deoxy-6-phosphogluconate, or KDPG, which then immediately breaks down into 2 3-carbon compounds called pyruvate and glyceraldehyde 3-phosphate, okay? So you have, and this is kind of unique, right? I mean, with the EMP, we saw formation of 2 G3Ps, right? And then we saw that both of those G3Ps would then go through the 3-carbon reactions to give you pyruvate. So here, you already have formation of a pyruvate, but the G3P that remains would undergo the same set of reactions that we already saw in the Embden-Meyerhof pathway, right? And it will still give you the NAD that we synthesize with the EMP, it will still give you the 2 ATP molecules, right? So you're producing a net of 1 ATP, 1 NADH, and 1 NADPH, right? With the EMP, we made 2 ATP, okay, net of 2 ATP and 2 NADH, and we did not make any NADPH, okay? And notice there's, there's no new precursors that are being synthesized here, but of course the pyruvate and the precursors that are in the 3-carbon stage are going to be the same as the EMP. All right, so the third pathway is the pentose phosphate pathway, and all cells have it, including our cells. And it's unique because it's referred to as a sugar conversion or transformation reactions. Okay, it takes glucose, it converts into various different types of sugars, so cells can remove those sugars from these pathways and use them for synthesis purpose, for anabolic purposes elsewhere. And because they're producing all of these intermediates to channel into anabolic reactions, they're also going to generate a lot of these NADPH, the source of electrons that those anabolic reactions also require. Okay, so we'll kind of follow this. You start off with 3-glucose-6-phosphate. The only reason that they're showing you that is because they want you to have these balance equations. Okay, but we can, look at it 1-glucose-6-phosphate. I'm not going to ask you to reproduce these. I'm not asking you to give me balanced equations, but that's the whole idea, that they're starting off with 3-glucose-6-phosphate so that you could count all the carbons and you could account them right? So that you're not like, well, we started off with, just 6 carbons, and how come we're ending up with all of these carbon compounds? And if I counted, they're like 18 carbons. Like, where are they coming from? So they're telling you, we're going to start off with certain numbers so that you could have a balanced set of reactions. So the first step is the same as what we saw, with the ED pathway, that it will eventually be converted into 6-phosphogluconate, and then the reactions beyond that are going to be, somewhat different. Okay, so those that you're seeing in these blue arrows are unique to pentose phosphate pathway, and then beyond this is essentially, notice it's even written here, it's going to be the same as the first pathway that we talked about, the EMP, conversion of glyceraldehyde 3-phosphate into pyruvate, right? So really kind of focused here. So once you have the conversion of 6-phosphogluconate into a 5-carbon ribulose sugar, okay? Obviously, if you went from 6-carbon to a 5-carbon compound, then you must have lost a carbon. So for each sugar, you lost 1, so you have 3 carbon dioxides that are produced. Now, this is an important intermediate because it's a 5-carbon intermediate. It can be converted into its isomers, xylulose and ribose, and it's the ribose is that 5-carbon sugar that is required for synthesis of nucleotides that are needed for RNA, right? mRNA, tRNA, and ribosomal RNA and DNA, right? So we have that ribose sugar, and if a microbe is going to rely on its own machinery to synthesize nucleotides, then it needs to make ribose as well. So here, this pathway allows a microbe to grow on a 6-carbon sugar source, but convert it into a 5-carbon sugar, which then it can use to synthesize nucleotides. And then it has 2 major enzymes that carry out the remaining conversions, the transketolase as well as the transaldolase. The intermediate that can become the ribose is going to be the 3-ribulose 5-phosphate. Okay? All right, so these reactions are noted where the individual enzymes are active. So you have then the transketolase that will react the ribose and the xylulose together to give you, those are 10 carbons. You still have 10 carbons, glyceraldehyde 3-phosphate, and a 7-carbon carbohydrate called the acetoheptolase. Okay, so you have the glyceraldehyde 3-phosphate, which can still be a intermediate for synthesis of amino acids as well as the lipids, because you have that intermediate. And then you have the transaldolase enzyme that is going to take those 2 carbons and then convert them into a 6-carbon sugar and a 4-carbon sugar. So fructose 6-phosphate, we can, convert it back into glucose, or we can use it to synthesize peptidoglycan, or we can use it to synthesize ATP if we wanted to. So we can, or just store it as glycogen, whatever the cell needs at the time. But, erythrose 4-phosphate is kind of new here. So erythrose 4-phosphate is going to be the backbone for synthesis of aromatic amino acids and vitamin B. Okay, so things like phenylalanine and tyrosine and tryptophan, right? These require the presence of this erythrose 4-phosphate. And of course, there's a series of steps after this, after it's been pulled out of here, to make that aromatic ring and all of that. And I'm not going to get into any of that, but this is going to provide that first step for microbe to be able to at least channel it into the synthesis of those amino acids. So microbe could be making this, but if it doesn't have the remaining enzymes to make the aromatic amino acids, it will not be able to do so. Okay, and so what is then it's going to do with the erythrose 4-phosphate? The erythrose 4-phosphate can combine with the xylulose 5-phosphate, which was derived from this sugar at the third step, to undergo a reaction catalyzed by transketolase to make you fructose 6-phosphate and glyceraldehyde 3-phosphate. So things that we know what to do with because we have the EMP pathway that we can channel it through all the same reactions that we have with the first glycolytic reaction. So really, it's these steps that sort of are different. Okay, the remaining steps are essentially the same with the EMP pathway. Okay, any questions that you have?
5 carbons. And then we go into the ribose 5 and then xylulose 5
right.
carbons. So that's 10 carbons. So where are those coming from?
So these are, remember, you have the 3 5-carbon compounds. Yeah, so now you have 3 of them here. 1, 2, 3. 3 arrows that you can count to make 15 again.
Okay. Thank you.
Sure. Anything else? Just follow the arrows, guys, and just understand the intermediates and what they're used for. Okay? You don't need to, I'm not going to ask you to reproduce any of this. Okay? I just want you to understand what they're using it for. Okay? They can either use it for synthesis, pull it out from these series of reactions, or keep it for catabolic reactions and make ATP. Okay? It depends on what the cell needs at that given moment. All right, so you have, what happened? So we have pyruvate, okay? At the end of each of those 3, whether we were working with EMP, with ED pathway, with the PPP, it doesn't matter. At the end, if it's catabolic reactions, we're gonna end up with pyruvate one way or another, okay? And so if the cell was gonna undergo aerobic or anaerobic respiration, it would convert that pyruvate into a substrate for TCA cycle. Okay, so here you have the chemical formula for pyruvate. Again, I'm not interested in you memorizing the formulas. If you know the names, we're good. So you have the pyruvate, which then has to be converted into a substrate for TCA. So pyruvate would be converted into acetyl-CoA. Pyruvate was a 3-carbon compound that we have at the end of each of the glycolysis reactions. And it's going to undergo 2 set of reaction, a dehydrogenation reaction, which means that hydrogens would be lost. And when you're losing hydrogens, you're being oxidized and you need to reduce a carrier. So you have NAD that is being converted into reduced NADH. Okay, so that's one reaction. The second reaction is that one of the carbon is being lost to produce CO2, so you have this decarboxylation reaction also going on as well. All right, so there were 2 pyruvates. They would be converted into acetyl-CoA by combining a coenzyme CoA using a high energy bond, and that is what really prepares it for the next series of reaction. If this can't happen, if pyruvate can't be converted into acetyl-CoA, then it can't enter TCA it's kind of is going to have to be channeled into fermentation, okay, which we're going to talk about in a minute. So you have, three reactions going on here: conversion of pyruvate into acetyl-CoA. A dehydrogenase called pyruvate dehydrogenase is involved that carries out all of these reactions. It will also strip off hydrogens, and it will reduce an NAD, and it will also result in loss of the carbon dioxide. So this reaction occurs twice because you started off with two pyruvates, right, the products of the EMP. Or if you started with, it doesn't matter, we'll keep with the EMP. And so you started with two, and so you will have these reactions occurring twice. You have twice the acetyl-CoA. Now, acetyl-CoA is prepared to enter TCA cycle, but it must combine with a substrate, okay, before it could become a proper substrate for the TCA cycle. So it must combine with a 4-carbon compound called oxaloacetate to make citric acid or citrate. Okay, so citrate is just the salt version of citric acid. Okay, so it's the same thing. And you have the conversion of citrate into isocitrate or isocitric acid, which is a isomer of that. It's just rearranging the atoms within that same molecule, making it more likely to enter the next series of reactions, which are going to be both, notice, dehydrogenation, so you have the NADH forming, and decarboxylation and CO2 forming. Okay, so you have two of these steps where first isocitrate is converted into alpha ketoglutarate, and that is coupled with dehydrogenation and decarboxylation. And then you have the alpha ketoglutarate being converted into succinyl-CoA, that is also coupled to those two reactions. So if you started out with 2 carbons with the acetyl-CoA, at this point you have completely dismantled that original carbon skeleton, and the remaining reactions are really conversion of oxaloacetate back, okay, from succinyl-CoA back into oxaloacetate. That's the whole purpose of a cyclical series of reactions, that you're going to regenerate one of the substrates. In this case, we're going to regenerate the oxaloacetate, so succinyl-CoA will be converted into succinate, and that is coupled with the production of either ATP or GTP. In bacteria, mostly it's GTP, but this is a substrate level phosphorylation reaction. And then you have the conversion of succinate into fumarate, which is coupled to synthesis of a different carrier, okay, FADH2, okay, and so this is the only place where FADH2 will be produced during TCA cycle. And then you have the conversion of fumarate into malate, and conversion of malate back into oxaloacetate is also coupled with production of more electron carriers. Okay, so you can sit down and you can simply just count how many electron carriers are being produced here per acetyl-CoA, and because you had 2 acetyl-CoA, you can twice the number. So you have 1 here, another 1 here, so you have 3 NADH per acetyl-CoA, so that's going to be 6 NADH for the 2 acetyl-CoA, 1 FADH2 per acetyl-CoA times 2, and then you have 1 GTP per acetyl-CoA times 2. Okay, so that's the total sort of energy extraction from TCA cycle. And notice again, there's not much ATP that is being produced. Okay, and these series of reactions are only going to occur in microbes that are undergoing aerobic or anaerobic reactions. If they are not undergoing that, if they're undergoing fermentation, then they stop at the pyruvate step. That's it. They don't go beyond that in terms of TCA cycle and electron transport chain. Okay, now we have dismantled everything. We have made all the CO2. You can count the CO2s. We made, 2 CO2s during conversion of pyruvate into acetyl-CoA, and we made 2 CO2 per acetyl-CoA times 2, so 4 CO2. You started off with 6 carbons. By the end of TCA cycle, you have 6 CO2s. Okay, so you're done dismantling the carbon skeleton. Okay, so what are the rest of the reactions for then? The rest of the reactions are going to reoxidize your electron carriers NADH and FADH, right? So notice we have just synthesized those carriers. We haven't really used them to make ATP. So the next steps involve taking those carriers and then passing on the electrons that they have to a set of carriers that are located in the plasma membrane for bacteria. In eukaryotes, the carriers are in the mitochondria, on the inner mitochondrial membrane. Bacteria don't have it. Their electron transport is found at the plasma membrane. Okay, so it's important for bacteria to have a fully expanded plasma membrane because they really need, every bit of that surface area for various reactions. So when we look at NADH and FADH2, what we're going to notice is that NADH can be used to make more ATP.